Welcome to another episode of Meet the Researcher. Today, I'm excited to welcome VHLA Research Council member and 2019 VHLA Research Grant recipient, Dr. Betty Gardie from the Ecole Pratique des Hautes Etudes Research Unit of l'Institut du Thorax CNRS in the University of Nantes in France. Dr. Gardie is an associate professor and has been working on the VHL gene since 2003. Dr. Gardie, thank you for joining us today.
Thank you. Thank you very much. I'm very happy to share my research project. I'm very grateful to the VHL Alliance, and thanks to the association and its generous directors, we will be able to progress in our research program. So I will present you my project. So you will see the title seems complicated, but you will see at the end of the presentation, you will understand everything. So the title is New Insight on VHL Disease, Characterization of a Novel VHL Cryptic Exon, and Modeling of VHL-related disease with Human-Induced Pluripotent Stem Cells, differentiated into neural cells. So it's a long title, but you will see it's not so complicated. So, next one. How can I change the slide here? So let's start by oxygen. So the oxygen and the adaptation to low oxygen is a topic that has been recently rewarded by the Nobel Prize, as you know. And oxygen is an essential molecule for life. You breathe it, it enters into your body, and it goes through all your cells. And cells need oxygen to produce energy and to live. Therefore, when the concentration of oxygen goes down, when you are in altitude or you climb up a mountain, you trigger adaptation mechanisms. So what are these mechanisms? So one of the important mechanisms is the production of erythropoietin. So you may know this molecule, EPO, which is largely, unfortunately, used during doping for high-level sport as doping. And this molecule is really active to produce red blood cells. So this erythropoietin induces erythropoiesis. But also you trigger the upregulation of vessels production, so angiogenesis. We call the production of vessels, but also vasodilatation. So then you allow a better dissemination of oxygen through the body. You also increase the glucose uptake to feed your cells without oxygen, and there is induction of survival factors. So all these changes are due to specific factors named Hypoxiainducible factor. And this factor will bind DNA and activate genes to produce erythropoietin that induce red blood cell production, vascular endothelial growth factor, so VEGF, to induce angiogenesis, etc. But this factor need to be active only in the absence of oxygen, so only in hypoxia. So what happened in normoxia, normal condition of oxygen? And here you can see this factor. So there is two subunits of this factor and one alpha and one beta subunit, and I will talk about the major isoform if 2 alpha. And this factor need to be degraded. And indeed, in presence of oxygen, the deoxygenase PhD will modify this factor. And VHL will recognize the modified factor with oxygen and will destroy the if factor to inhibit the factor in presence of oxygen. So what happened in some cases? These major actors may be mutated in pathological conditions, and these mutations induce stabilization of the F factors, and then you have upregulation and overproduction of erythropoietin that induce overproduction of red blood cells, what we call polycythemia. But you have also increase of the vessels numbers or survival factors, and these mechanisms participate to the occurrence of tumors. So the first example of the gene involving hypoxia pathway was the VHL gene. The first disease was the von Hippel-Lindau disease, described in 1936. And the gene was discovered later in 1993. And here you have the representation of chromosome 3 and the location of the VHL gene. So every cell in the body contains 2 copies of each gene. So one inherited from the mother and one inherited from the father. And here, the von Hippel-Lindau disease is due to one mutation in one copy of the gene. And patients with one mutation are predisposed to develop multiple hypervascularized tumors. So here is the location of tumors from the eye of the body to the top to the bottom. So first you have retinal hemangioblastoma, endolymphatic sac tumors, hemangioblastoma all through the central nervous system, pheochromocytoma, renal cell cancer or cyst, and neurocrine pancreatic tumors or cyst, pancreatic cyst. And depending of the VHL mutation, there is some examples here, the severity of the disease is not the same, and some mutations, for example, only predisposed to pheochromocytoma, and other mutations predisposed to all types of tumors. But more recently, in 2002, another disease has been associated with VHL mutation. So this patient has been identified in Republic of ex-Russia, named Chuvash Autonomous Region. And these patients presented 2 mutations this time, one in each copy of the gene, a particular mutation named arginine 200 tryptophan, so R200W. And this mutation in both copies predisposed patient to overproduction of red blood cells. So the age at the diagnostic was very young, so from 6 to 20 years old. These patients presented polycythemia from birth, so what we call congenital polycythemia, with elevated level of erythropoietin. They suffer from headaches, vertebral hemangiomas, varicose veins, pulmonary arterial tension, and unfortunately, they die prematurely due to cerebral vascular events and peripheral thrombosis, so around the 50. But the characteristic of these patients is the total absence of tumors. So these patients do not develop tumors. So neither the patients nor the parents who are healthy with only one mutation, like the VHL patients. So if you look at the different VHL mutations associated with the different disease, you can see a very different disease associated with VHL mutation. So either the development of multiple tumors or development of polycythemia when there is 2 copies of the genes mutated, or patients perfectly healthy. So the question of the laboratory was to understand what are the molecular mechanisms at the origin of the different diseases. So what we show in a recent paper is that there is a correlation between the severity of the disease developed by the patient and the activation of the hypoxia pathway. So the more active is the hypoxia pathway, the more severe is the disease. And at the bottom of the gradient, you find polycythemia. So we wonder if there is other genes involved in hereditary hypoxia-related disease. And indeed, many laboratories, and we confirm this observation. And for example, polycythemia and pheochromocytoma and paraganglioma may be due to mutations in either PhD2 or the If2 alpha gene, but also mutations in the gene named FH for fumarate hydratase and METF can predispose patients to pheochromocytoma and renal cancer. They can also predispose patients to other disease linked to the skin, leiomyoma or melanoma for FH and METF. So the project of the laboratory was to understand these differences, and we think that to understand the VHL disease, we need to study and compare all these hypoxia-related disease. So the first step was to compare collection of patients, compare mutations, compare associated disease. And thanks to Anne-Paul Gymnase-Recoplo, who work on pheochromocytoma and paraganglioma, and Stéphane Richard, who follows 900 patients with the hippel-lindau disease, and with Marie-Françoise Avril, who work on hereditary skin lesions. We have been able to compare and start to study the different diseases. But a collection of patients with polycythemia was missing. So polycythemia are really rare genetic disease with no identified mutation in 80% of cases. And the hypothesis was that study polycythemia may be very helpful to understand the other disease. So thanks to François Giraudon, we collected 300 patients with polycythemia. We sequenced 250 from 2015. And we identified at least 20 patients with a VHL mutation. So even though it is a rare condition, we recruited enough patients to perform interesting studies. And the second point is to establish a disease model. So we need to be able to compare the mutation and to study this disease in a dish. So how can we do that? So can we use animal models? So the answer may be yes for polycythemia. So you can see here that researchers managed to reproduce the disease in mouse. So you have here a mouse with the equivalent of the VHL Chauvache mutation. And these mice present a higher hematocrit. That means they produce an excess of red blood cells. And maybe you can see here, they have a red nose, which is characteristic of overproduction of red blood cells. So this model perfectly reproduces Chauvache polycythemia. And if you look at the zebrafish model, you can see also, if you invalidate VHL in zebrafish, you can see an overproduction of red blood cells. So this can work for polycythemia. However, for the VHL disease, there is no perfect animal model, because the VHL mutation by itself does not reproduce, the mouse with VHL mutations do not develop all the tumors that we can observe in patients. So we decided to work with a human model. And we think it's really important because as a human genome is really complex, and maybe it's not always comparable to other animal genomes. So here is represented a chromosome that contains the genetic material. And if you go deeper, you can see here that only specific part, the red part, can be translated So there's different steps. And at the end, you have the active protein that is functional in the body. And if you look at the proportion of this here and of this exon here, you can see that it represents only 1% of all the genetic materials. So only 1%, so it's very little. And for example, in purple, so all the rest is calling non-coding sequences. And here, for example, the purple part, it's intron, which I represented here. Here is the introns. And this introns represent 26% of the genetic material. So an additional point is that the non-coding sequences play a crucial role and may be more complex in higher primate. So if you look at the difference between all these organisms, plants, worm, fish, and human, the coding sequence that produce proteins, if you compare them, they are the number of genes, or I mean the number of exons and the coding sequences that encodes proteins are similar. But if you look at the ratio of non-coding sequences, the more complex is the organism, and the higher is the number of the non-coding sequences. So we think that to work on human genetic disease, you need to pay attention to these non-coding sequences. They may hide something. So we decided to work on the human cells to study the human disease. So the best current available model to study human disease is what we call the stem cells, or iPS cells, for induced pluripotent stem cells. So we take cells from patients, either urine or blood, and we reprogram them to be able to obtain cells that you can then transform in all the cell type you want, either neural cells, muscles, blood, bones, etc. And so we will use these cells to study VHL disease. So now the question is, what type of cells are responsible for the hypoxia-related disease? So we know that, so we only work on hereditary disease. So from the beginning, I just talk about hereditary disease. And in that cases, all the cells carrying the mutations, so all the cells in the body are mutated, here in black. But the disease is only expressed in a few cell types. So polycythemia, I mean, overproduction of red blood cells, we need to know which cell types are responsible for this overproduction of red blood cells. And for the von Hippel-Lindau disease, we need to identify the cells at the origin of tumors. And we have a working hypothesis. So it's just an hypothesis, but I would like to share this with you. So we think that all the cells at the origin of this different type of tumors, these cells have the same embryonic origin. And this embryonic origin is what we call the migrating neural crest cells. So let's see in details, what are these cells? So let's start with the polycythemia. So during development, you have here the cells that produce erythropoietin. So you can see erythropoietin in green. And cells that produce erythropoietin are present in the retina and all through the spinal cord. And these cells are migrating neural crest cells. Hepatocytes also produce EPO. And these cells, the migrating neural crest cells, will then migrate and all through the body. And they will end notably in the kidney. So you can see here in blue, the cells originating from neural crest. They are in blue here. And you can see also that erythropoietin in green in the kidney here, and more closely here, the green cells In fact, when you overlay these two pictures, these cells are the same. So we know that interstitial cells that produce erythropoietin in the kidney originate from neural crest. So no doubt for this disease. Now, if you look at tumor development in patients with von Hippel-Lindau disease, we also think that there is only one single cell type at the origin of all these tumors. First, if you look at the migrating way of these neural crest cells, you can see retina and all through spinal cord. So what are these cells? So these cells, they emerge from neural crest during the development, and then they migrate all through the body and they become cells with specific functions. So, for example, they become neurons, glial cells, so sorry, it's in French, surrenal glands, etc. And these cells are very good candidates to be at the origin of hypoxiarelated tumors. So endoplymphatic sac tumors in the ear, pheochromocytoma in surrenal glands, hemangioblastoma all through the spinal cord, but also for FH and MTF gene, like we saw before, melanomas and leiomyomas, which emerge from cells from muscle cells. In French, muscles. So sorry for the translation. But there is a debate regarding the origin of cells from which give some hemangioblastoma. Indeed, the tumors from hemangioblastoma are vessels tumors. But also there is a debate regarding, there is no debate regarding renal tumors, because we know that tumors of the kidney come from epithelium kidney. So what is the link with the neural crest? And indeed, it's like if you look here at the origin of a messy stuff. So here is an example of a tumor that accumulates multiple damages. And sometimes you may need to look up. So what I mean, it's like entering in a bathroom full of water and floating bottles. So you may want to put back all the bottles, identify them, and tidy things up. Or maybe you just want to stop, to stop the crazy little monster who is playing with the water. So what I mean is that in research in cancer may pay attention to the environment of the tumor, and maybe there can be an indirect effect. So our working hypothesis is that tumors emerge from migrating neural crest cells. So for example, hemangioblastoma, cells all through the spinal cord originating from migrating neural crest cells can produce angiogenic factors, and then vessels proliferate and induce hemangioblastoma. And for the kidney, these migrating neural crest cells migrate into the kidney. And if there is mutation and a weak dysregulation, so here you can see the erythropoietin is produced, and then you have overproduction of erythropoietin and polycythemia. But if you have a strong deregulation of these cells may be sick and produce inflammatory cytokines or produce toxic factors that can be deleterious for the neighboring cells in green here that are at the origin of the tumor. So thanks to the VHL Alliance, we will be able to use this model of IPS. So we will establish IPS cells from patients carrying different mutations, so either polycythemia or von Hippel-Lindau disease. We will differentiate them in neural crest crest cells. So here is what we have in a dish. And we started our study first with a patient with a Shubash mutation, and you have here the first results, and you have the erythropoietin level we obtained, and you can see in red that indeed we managed to produce EPO, because the erythropoietin is only few cell types produce erythropoietin. It's very, very difficult to force cells to produce erythropoietin, and here we managed to produce erythropoietin from our cells. So now we will compare different iPS cells and compare the factors they are expressing and maybe identify therapeutic targets. So these experiments take a lot of time, because for each patient, 6 months are needed to obtain the iPS. And then for each mutation, you need to reproduce the experiment 3 times and 2 different clones, etc. So it's very time consuming. So now let's continue and finish to the third point of the project, is the identification of the molecular mechanisms. So we need to know what happens in different pathologies. So I told you that it was very important to work on a human model, because we hypothesize that the human VHL gene is indeed more complex than all this previously described. So here is the structure of the VHL gene. It's located in chromosome 3. It has been described as a very simple gene. So 3 parts of the gene will produce the VHL protein. So if you go deeper, you can see here. So these 3 exons can combine in 2 ways. So the major combination is exon 1 combined with exon 2 and exon 3. And this produces the functional VHL protein. So 2 isoforms, a short one and a longer one. And these 2 types of proteins are able to inhibit IF. So the main action, the main function of VHL. And there is another combination. So exon 1 combined with exon 3. So it's a very minor isoform. And this produce another protein, which we still don't know its function. But what we found recently, that was really surprising, because indeed we find that the VHL gene is more complex than we thought. And it contains what we call the hidden exon, named exon 1 prime. And this exon 1 prime is located in the non-coding region. So this, like I said previously, this non-coding region are more complex in human, and this exon 1 prime is only present in higher primates. So we can work on this exon only in human cells. So this hidden exon, E1 prime, can be combined with other VHL exons. So either it combined with exon 1, exon 2 and 3, or exon 1 and 3. And also it can be produced by itself and combined with exon 2 and 3. So we still don't know if this can produce active protein and what are the functions. But what we know is that this exon 1 is really important for the VHL disease. And indeed, so we identified this exon thanks to study of Polycythemia. And what we found is that we identified families, 7 families, with mutation in E1 prime. So the 6 first family have been identified and arrived in our laboratory because they had only 1 mutation in VHL. So it was classical Shubash mutation for the family 2, 3, and 4, or other mutation in other classical VHL exon. But we knew that polycythemia is due to 2 mutations. So we looked for the second event, the second mutation. And we found this second mutation in this hidden part of the gene. And for the last family, it was a family with a polycythemia really close to the one observed in VHL patients. And indeed, the patient inherited from one of each parents of 2 mutations located in E1 prime. But more interestingly, in the laboratory, we worked on a large family with one hippel-lindau disease and absolutely no mutation, neither in exon 1, exon 2, or exon 3, nor in the promoter, or no mutation at all. But clinical manifestation really typical of the VHL disease, so hemangioblastoma of CNS and renal cell cancer and phlebochromocytoma. And we found mutation in this exon 1 prime and in all patients presenting the disease. So what does this mutation in E1 prime? So thanks to our institute, we had a very performing next generation sequencing platform. And we can sequence very deeply all the cells. So we sequence patient cells. And here, then you can see how these exons are expressed in these cells. So here located exon 1, exon 2, exon 3. The peaks represent the expression. So the more high is the peak, the more expressed is this exon. And the links here are all the combination possible between the different exons. And what we see that in patients, so no, first, in the control, in the green control, you can see that exon 1 prime is expressed at a very low level, but can be expressed. But mutations in E1 prime, either in blood patients or in phlebochromocytoma, here, you can see that mutation induce an increased expression of E1 prime. So this exon 1 prime is included in the gene. So we still don't know the consequence. So our hypothesis is that this dysregulates the production of the functional combination of exon. I mean, exon 1, 2, 3 are functional. And then you have less production of functional protein, VHL, and then you have less VHL able to dysregulate if. But we still don't know what is really the consequence of this mutation. So we are still working on it. So in conclusion, we show that the VHL gene is more complex than described in 1993. We show that study of polycythemia can be informative to understand the VHL disease because we identified this hidden exon thanks to study of polycythemia. And we think that the study of cells at the origin of this different disease is the key point to identify the mechanisms responsible for the pathogenesis and for the identification of novel therapeutic targets. So thanks to the VHL Alliance, we will be able to work on this new VHL E1 prime in order to identify its role. So we will study patients with mutation in E1 prime. So we have a collection of patients. 18 patients have been identified with the causal mutation. So 10 are still undescribed, we need to work on it. We will establish iPS cells from patients carrying selected mutations. We will differentiate them into neuron crest cells. And we will study the genetic and the genomic of these cells by using RNA sequencing in order to know the expression pattern of these exons, the impact of the mutations, correlation with the disease severity, and we hope to identify the dysregulated pathway in order to identify the new therapeutic targets. So I would like to thank all the people working with me in the past and in the future. So the VHL Alliance will help me, but also the group of Yannick Arlot in Rennes with Franck Chanel and Anne Couturier, and also Stéphane Richard and Sophie Couvé. We will help to work on this project. So I'm really happy now to share your questions.
Dr. Gardie, thank you so much. That was wonderful. We have a number of questions for you. The first question is, how did you become interested in research into hypoxia-related diseases, including VHL?
Okay, so I've been recruited in a Stéphane Richard laboratory, and he built the survey of patients with VHL disease, and he offers me the opportunity to go to my first international meeting on VHL. It was in Kochi in 2004. And there I meet fantastic researchers and fantastic patients also that really motivated me to work on this disease. Really interesting. And meet patients, I think, is the key for a researcher to. Now I know for who I work.
Wow, that's really important. I know that patients, it's important to patients to actually get to meet people who are doing the work and this great research. So the next question is, why should the general population support research into these hypoxia-related diseases that you describe?
Why? Sorry, can you repeat?
Why should the general population support research into these types of hypoxia-related diseases?
Why should, or why should? Yes, and indeed, oxygen, as I said, is the molecule of life. And it involves more than what I discussed today. It can modify all the behavior of the cells, and it involves also adaptation to live in high altitude. So everybody is concerned when you go up into the mountain. You may be sick due to hypoxia, but also hypoxia, as I said, induce survival mechanisms. So it's a really major factor that induce tumorigenesis, not only hereditary tumorigenesis, but tumors in general. And this is really a pathway that needs to be targeted in cancer for everybody. All the cancer are concerned of this topic.
So the next question is actually just what you just said is, does living in high altitude areas have any impact on tumor development?
This is an interesting question because I don't have the answer. What I know is that the first mechanism is the induction of red blood cells. So the disease of altitude is overproduction of red blood cells. And people who are used to live there from thousand years, they have adapted their hypoxia pathway, and they can manage with the production of red blood cells. But tumorigenesis is a very long process and your body adapt, I think, when you live in altitude, and I'm not sure it can influence tumor genesis. I don't have the answer, but it's a very good question. Very interesting question, yes.
Are patients with VHL more susceptible to getting melanoma?
I don't know. I mean, I'm not a doctor, and I don't think so. I don't think so. The migrating neural crest cells, they, during the development, they acquired different properties to, at the end, become very specific to a function. And I think mutation in VHL have an impact on cells that will become surrenal glands, etc., but not at the end of the production of melanocytes. See what I mean? So there is no predisposition of patients with VHL to melanoma. No, this is sure.
The next question is, how will the identification of the molecular mechanism of VHL impact the lives of VHL patients and families?
We are looking for new targets. So it's important to stop the tumors when they are developing, but I think it's important to avoid them to start. And we are looking for the very, very first step of the emergence of tumor. So we need to know what factors, what are these toxic factors that these cells may produce, to be able to identify them and then find a therapeutic target.
And then the final question, Dr. Gardie, is what do you think the VHL research landscape will look like in the next 5 to 10 years?
I hope we will be able to have a model, and first, and then to be able to survey patients. So to have a very good correlation between the mutation and the risk to develop some tumors, and then to be able to survey patients and be able to say, okay, you have this type of mutations, you have no risk of developing renal cell cancer or etc. And to be more precise on the prognostic, and I hope, of course, more therapeutic targets to be tested and efficient on patients to cure the disease.
Dr. Gardie, one final question. Is heart disease or ischemic heart disease related to VHL at all? Or is there any indication of that in the research?
I don't think so. No.
All right. Once again, Dr. Gardie, I just want to thank you for taking the time to join us this morning, and especially for all the important work that you're doing to advance VHL research and knowledge. I know that the VHL Alliance appreciates it, but also the VHL community and the greater community just very much appreciates your work and for you taking the time to join us. So thank you so much.
Thank you to you. I'm very grateful of your support. Thank you very much.
Thank you. And for those watching at home, you could find a calendar of future Meet the Researchers, as well as other events in the Surviving and Thriving Together series, in addition to video recordings of all past events at vhl.org/together. Until then, stay safe. Dr. Gardie, thanks again.
Thank you.